So, I am Daniela Cirillo. I work at the Università Vita-Salute San Raffaele in Milan, Italy. My position there is the head of the Emerging Bacterial Pathogen Unit, and I'm responsible for the Clinical Microbiology Unit. Within the UNITE4TB, I'm leading the microbiology work package. Yes. In terms of new diagnostics, we have now more tests than in the past. So we have more molecular tests, but we are still very far from having ideal tests, and I will tell you why in a few minutes. Regarding the new treatment, we are also making big steps forward because now we have shorter regimen and more friendly because they are becoming all oral, also for patients with drug-resistant tuberculosis. In terms of what is needed, we are still far from what is needed, especially in terms of diagnostics that are able to detect in very short time the people that are going to fail to the treatment, both in terms of biomarkers for prediction of failure, but also for capacity to make the diagnosis of drug resistance to the new drugs. The diagnostics that are today commercially available were designed based on older regimen, in particular for patients with drug-resistant tuberculosis. So we are not there yet. In addition, we need to move closer to the patients with our diagnostic capacity. And that can be done with two strategies, either having tests that could be performed very close to the patient location, may be able to be done in a very decentralized manner, or having a very strong capacity to collect and centralize samples and running the test in a more centralized manner. Today, both of these strategies are not perfect and are not ideal to support the diagnosis of tuberculosis, especially in high burden settings. So the main diagnostic gap that we see is the capacity to provide a rapid detection of resistance to the particular drugs that are used for treatment. Tuberculosis bacteria, they grow very slow in culture, so the strategy that we need should be faster than the growth rate of bacteria. So we need to focus on our molecular capacity to identify drug resistance. In order to do so, we need to develop capacity to do genome and identification of the mutation that are causing resistance to the drugs used for treatment. Regarding the drugs, the main gaps are the side effect and the cost of the therapy. Side effects are still very relevant. We are now moving to a fully oral therapy, also for drug-resistant tuberculosis, but still the therapy is longer than we would like to have, and there are people that are either failing therapy because they develop drug resistance, or they cannot complete the therapy because there are important side effects. So we need to simplify the therapy, and we need to make the therapy very user-friendly. UNITE4TB can really accelerate the use of new diagnostic in high burden setting showing the importance of new diagnostic and helping to this process of co-development of new diagnostics that are coupled to the new drugs and drug regimen. What is needed is showing the impact of new genomic-based diagnostics or biomarker-based diagnostics to predict the treatment outcome very early and support the decision by the clinician to address the patients toward the proper regimen. We are giving high importance to comparability across the different trials that are going on. We make sure that our standard operating procedures are comparable to those used in all the other trials. We would like to standardize also the microbiology component in all the trials in order to collect the data that are needed to compare the results of the trial from this aspect over time for the 2 days ongoing trials and for the future ones. That will really speed up our capacity both to improve on the diagnostic needs and also to understand which kind of resistance may be developing during the trials and make us in the position to address this.
So let's MDT together.